DNA structure. It's structured by a double helix, which is a structure formed by double-stranded models of nucleic acids. This is a DNA nucleotide. It contains a phosphate group and a nitrogenous base, or a deoxyribose sugar. This is a nitrogenous base. These two complementary nitrogenous bases are bonded by a hydrogen bond. This is a sugar-phosphate backbone. And together, it's bonded by a covalent bond. This whole thing is a DNA strand, and this is a purine and this is a pyrimidine.
So this is the DNA replication process. So first comes the topoisomerase, which is right here, and it's an enzyme that untwists the backbone. Then comes the DNA helicase, which is an enzyme that unzips DNA by breaking down hydrogen bonds between bases, which creates the replication fork. Then comes the single-strand binding proteins, which bind near the replication fork and hold DNA apart so hydrogen bonds don't reform. Then comes the RNA primase, which are enzymes that lay down on an RNA primer, and a primer is short sections of RNA, which are like right there, right there, right there. Then comes the DNA polymerase 3, which are right there, right there, right there, which are both found in the leading and lagging strand, and they both add DNA nucleotides, which are adenine, thymine, cytosine, and guanine.
Okay, these are the DNA nucleotides, which are these, and they are newly synthesized nucleotides. This is the leading strand, which is the blue strand here, and it runs from 5' to 3', and when it replicates, it runs from 3' to 5', and it is continuous, and it requires DNA polymerase 3. This is the lagging strand, which is this bottom strand, and it runs from 3' to 5', and replicates 3' to 5', and it's not continuous. It requires DNA primase, DNA primer, DNA polymerase 3, and DNA polymerase 1 along with DNA ligase. And then DNA polymerase 1, which are these, they remove RNA from primer and replace it with nucleotides. And then this is DNA ligase here, and it's the glue that joins Ogasaki fragments.